I'm glad to have an opportunity to introduce Juan. Um, Juan is a plant developmental and molecular biologist with a strong interest in crop improvement for sustainable agriculture. Juan graduated from the National University of Rosario in Argentina with a degree of biotechnology and later with a PhD in biological science. In 2014, Juan joined the lab of the Professor Dubovsky at UC Davis in California, where he initiated as a human frontier science program, a postdoctoral fellowship, and later as a research specialist of the Howard Hughes Medical Institute. With Prof. Dubovsky, he led a group focused on understanding the genetic network that control wheat plant architecture and flowering time, and they affect the yield. Currently, Juan is a project scientist at the Plant Transformation Facility at UC Davis, where he lead the project focused on developing technology that improve gene editing and crop transformation. So the presentation title of Juan today is Boosting Plant Regeneration Accelerate with Improvement with Genome Editing Technology. So Juan, you can start when you're ready.
Okay, well, hello everybody, to those that just connect, and thank you, Sebastian, for the introduction, and thank you for the invitation. I mean, I'm happy to present my work in this seminar because I feel like part of the work that I'm going to present is kind of relevant, or I got many ideas and interest from your work that have been done in barley. With similar genes and similar mutation you will see later. So I'm happy to be able to show those data here. And I organized the seminar in two sections. A first part, I will introduce and describe the technology that we developed with Jorge and David Tricoli that help us to transform with high efficient wheat and other plants. So I'm more or less I'm going to talk how we come out with that. So that's the project that I started actually in Argentina during my PhD. And in the second part, I will show an example of how this technology can help us to move faster from ideas or hypotheses to testing those ideas in a cultivar that is already elite and in the field. I will switch to the presentation here. So starting from this first part, so I like to say, or I did my PhD in Arabidopsis. So for me, plant transformation was always an essential tool for research. When I moved to working with Jorge for my postdoc in wheat, I was always trying to improve and test my hypothesis using transgenesis, because I feel like that's a super powerful technology that can help to test ideas in an isogenic background. So I really like that, and it's an excellent complementation to genetics, but also with the development of the gene editing technology, that's more or less happened when I joined Jorge for my postdoc. So those two technologies are, again, super powerful for research and crop improvement. But the problem that you may know when you want to use these technologies is that plant transformation and regeneration, I mean, are main bottlenecks to apply them for crop improvement or for research. The main reason are because most of the cultivars or most of the crops are very hard to transform. The efficiency of transformation is low. There are a lot of genotype dependency, and the protocols that we normally use to generate those plants, transgenic plants, are very labor intensive. They require space and special laboratory space. So when I came to work with Jorge, there was a need to improve the wheat transformation, because this was a typical transformation in wheat that we have in the UC Davis plant transformation facility at that time. So the efficiency was too low. We were recovering 1 or 2 plants per experiment. So the number of events that we were getting was too low to do something really useful. So I started working with David Tricoli, that at that moment was the manager of the plant transformation facility. And our idea was to incorporate, so David has a lot of experience on tissue culture and all the protocols that involve hormones, treatment, stuff like that. But my background was more in molecular biology and plant development. So I started with David to combine tissue culture with what we know from plant regeneration and from a biological or cell biology perspective. So in this slide, I'm basically showing the basic of the plant regeneration. What we need to do is convert a fully differentiated cell, somatic cell, into something that is dedifferentiated, that can proliferate to make a calli and acquire potentiality. And from that cell, then we are going to regenerate embryos or organs that will be that if we transform them, those will be the transgenic event or the gene editing events. So all this year we have get a good understanding of the molecular network and the genes that act during this process. So there are many genes that right now are known that participate and can potentially be useful to improve the system. So example include Bushel-like genes, plethora genes, baby boom. So there are many genes that can be used to boost the regeneration or to promote the somatic embryogenesis process. But something that happened when we would start working with that, and it was very clear that it was that when you overexpress many of those genes, or when you have a constitutive expression of them, the problem is that you can induce somatic embryogenesis or gynogenesis on the explants, but the problem is these genes will keep the tissue at that stage or with a pluripotency identity, so they cannot differentiate longer or continue to grow as a normal plant. So there are a lot of developmental defects. But with David, we were lucky on finding that a transgene that we introduced into wheat was good to improve regeneration. And what it was very interesting about this technology, it was that it didn't introduce developmental defect. So the technology we call GRF-Shift. So I'm going to tell a little bit about those genes before I continue. GRF and Shift is two families of transcription factors that I worked during my PhD in Argentina. GRF stands for growth regulating factors. So these are plant-specific transcription factors that are highly conserved in plants. So you can see they are present in almost every land plant. And they were discovered many years ago by the group of Hans Kendi in the University of Michigan. But after Hans Kendi work, there has been a lot of work showing the role of those genes in plant and in plant growth particularly. And we were interested in characterizing those genes because they are the target of a family of microRNA, that is a microRNA 396. And Javier Palatnik, that was my previous PI, was very interesting, or he was part of the different groups that were studying microRNA activity and biology in plants. So we were very interested in understanding the connection between this microRNA and the GRF. I didn't mention that the GRF are characterized by the presence of these two domains, the QLQ domain that is involved in protein-protein interaction, and the WRC domain that is it participates in DNA binding. So it provides the DNA binding specificity to these transcription factors. As we were interested in understanding the biology of these genes, we working in Arabidopsis. For example, we check the expression pattern of them. We observe that the expression overlap with the region of the organs. In this case, this is a leaf primordia. The expression pattern overlap with the region that are undergoing cell proliferation. But what we also observe is that this expression pattern is the result of the activity of the promoter of the GRF, but also the activity of the microRNA. because, for example, this is a CRF reporter where we introduce point mutation in the microRNA binding site. So this is insensitive to microRNA activity. So these two have the same promoter, but you can see here that the expression is almost constitutive in the leaf primordia. And this is a reporter for the microRNA expression. So you can see that the microRNA is cleaning the CRF expression in those regions that are associated with the cell expansion and differentiation. As I say, the QLQ domain on the CRF mediates interaction with another gene family, protein-protein interaction with another gene family. This is called CRF interacting factor. So this is SHIF. And these SHIF are small proteins. It's a small family, sorry, that they don't have a DNA binding domain, but they can interact with chromatin remodeling complex and also with CRF. And the hypothesis is that they act as a co-activator of the CRF to control the expression of downstream or target genes. Well, okay, about the biology or the role of these genes, one of the first things that we did was to study them, was to knock down the expression or knock down the CRF by overexpressing the microRNA, or knock out SHIF by mutation. So, and you can see when you reduce the expression of these genes, the plants are smaller. So these genes, they are expressed in a region that overlap with cell proliferation. And if you reduce the expression, at the end you have smaller organs. And you can see that that's because there is a reduction in cell proliferation. So these genes are positive regulators of cell proliferation. But what is interesting also is that this role in cell proliferation not only affect organ size, but it can have also an effect on meristem homeostasis, because when we reduce even more the activity of this complex, for example, by crossing a mutant in SHIF1 and the overexpression of the microRNA 396, we can get plants that lack the shoot apical meristem. So this is showing that cell proliferation or the activity of these genes is also important for meristem homeostasis. On the other side, we also observe that if you overexpress these genes, for example, in these cases, by making a version that is insensitive to the microRNA activity, the plants are larger. And if we co-express this with SHIF, we can have an enhanced activity of CRF and get larger organs. But at that time, when I was doing my PhD, we also observed that in addition to the regulation at the transcriptional level and at the post-transcriptional level by the microRNA 396, the assembly of the CRF SHIF complex was also, we had the hypothesis that can also be regulated, because we knew at that moment, and now there are some publications, that there are other proteins that can interact with CRF and can compete with this assembly, complex assembly. So we had the idea of combining these two genes in a single polypeptide with a small bridge, and express them together as a single protein. And what we observed at that time was that when we put them together as what we call CRF SHIF chimeras, we can get a higher activity in this case in CRF3, and that was associated with an increase in cell proliferation. So the summary of this part will be that we were able to generate a single transgene that can be useful or super useful to increase cell proliferation very easily just by adding this transgene. And so going back to the regeneration problem, I told you that so this is the process that involves cell differentiation, acquisition, and new fate, but there is also a process of cell proliferation. So when the cell differentiate and need to acquire new identities, so cell proliferation is important, and then cell proliferation is important to establish those meristems that are being produced. So we have our CRF SHIF, and we transform that in wheat. So this is also, but to be honest, this was a huge surprise. So when we transform that in wheat, we have a huge increase in the efficiency of transformation. So I say that this is a surprise, or this was a surprise, because I mean, we were expecting to see an improvement on the transformation efficiency, but we were not expecting to see this difference. So this is like a day and night. So it was completely or a huge increase in the efficiency of regeneration. So we were a little bit skeptical at that moment. So David, that has a lot of experience on tissue culture, told me, no, we need to repeat this many times. So we repeat the experiment multiple times to be sure. Well, the data actually show that yes, it was good. We can improve regeneration, but also when we doing all these new experiment, we learn many other things like the process in general of transformation was much faster. So shoots were originating earlier and then they grew faster and quickly. So we could reduce the transformation process from 90 to half to 50 days, around 50 days. And the other thing that was very interesting is that in wheat transformation, there are a lot of restriction or condition that can limit the transformation efficiency, like a plant growing condition, the embryo size. And there is one step, for example, that is removing the embryo axe or the that's embryo section during the transformation that can be very laborious, but is relevant or critical for the transformation efficiency. But for example, using CRS shift, our technology, we were able to recover a very high efficiency of transformation, even without embryo excision. So you can see here, many experiments in the control were close to zero, where with the chimera, we were always above 30%, 40% efficient transformation. The other thing that was important was that the plant that we got after this transformation, they were normal, they didn't have deleterious phenotypes, and they produced it. So the tool seems to be very good. It's a single gene. And the first idea that we had was to combine this with CRISPR. So because we were able to put everything in a single tDNA, so Cas9 plus the CRF shift. And as a proof of concept, we test this with gene Q, that is a famous gene in wheat that is related to spike development and the free threshing character. But the knockout of Q in durum wheat produce a very clear and distinct phenotype that is this indeterminance in the, so it's already indeterminate the spikelet meristem, but produce more floret per spikelet. So we transform that, we generate in a single plate multiple independent edits, and when we transfer them to soil, all of them show phenotypes. So this was our first experiment combining CRF shift with CRISPR, and we could easily show that we can increase the frequency of editing events and that we can get. I mentioned now something that you may be interested in. It's something that we haven't published, but we developed that. So combining CRF shift with Cas9 is a very easy and a good strategy to do gene editing plants, but many times, and now that I'm in the plant transformation facility, researchers are interested in transforming single genes, not doing gene editing. Just, for example, if you want to make a reporter, or if you want to overexpress some candidate gene. So in those situations, it's good not to have CRF shift in the same tDNA, so you can somehow work with your gene alone. So for those situations like those, what we were doing, and now it's working very well in our hands, is co-transforming a vector that is a regular vector, we call, could be a reporter or whatever, co-transforming that with a vector, another agrobacterium that has CRF shift, but without the hydromycin cassette. And doing this co-transformation and playing with the dosage of the different agros, we can get now a very high efficiency of transformation for regular vectors. So that strategy is working very well for us in the facility, and now we can provide the transformation service that deliver a good number of transgenic independent events that you can do, or PIs can do research on that. Okay, the other thing that was very interesting on this technology that allow us to expand the range of genotype that we can transform. So initially I showed you Kronos, but we also transform a breeding line from our program that is called Desert King, but also we were able to transform other cultivars that in our hand before were recalcitrants, like Cadenza or Han. Cadenza is very interesting because the exaploid tilling population that Jorge and Cristobal sequence is in Cadenza, so having this tool is good to complement the tilling population. But also this year we published, I published in collaboration with CIMMYT, a work where we showed that, for example, CIMMYT was able to transform 6 cultivars that are relevant for CIMMYT and for farmers. So that's great for us. I mean, it's nice to get that working. More recently, so we were working also in translating this technology to other crops. And we have this collaboration with the group of Andrea Galavotti in Rutgers in New Jersey. Andrea works in maize, and he was interested on using this for maize transformation. Andrea, when he tried GRF shift alone, he was getting some increase, but it was not too much. So he wasn't happy enough to, or the efficiency was not high enough to be useful. But Andrea was also playing and working with Baby Boom, with the construct or the cassette that was developed by Corteva. And we have this idea of combining GRF shift with other morphogenes because we know that these genes don't act on the same pathway, so there can be synergism between them. So Andrea tested that. He combined Baby Boom with GRF shift, and he was able to get a very high and nice efficiency in maize. So here, this is a big table showing a lot of independent events, but the message is here. So this is efficiency with single genes like Baby Boom or GRF alone, and this is the efficiency when he combined them. So now, let's say this vector is already available in that gene, according to Andrea, is quite simple or more simpler to do gene editing in maize and in different genotypes, not just HI2, but also B104. So I will summarize this part. That is the first part that I told you at the beginning. So we developed a GRF shift tool that increases the efficiency of transformation. That technology results in plants with normal phenotypes, can be combined with CRISPR to produce high frequency of editing events, and also help us to expand the number of wheat genotype that can be transformed. So for me, this is a summary of GRF shift as a tool. Now I want to provide an example of how we use this new tool to test and introduce new genetics resources to control plant height in wheat and in triticale. So this project actually started when conversation that I normally have with Josh, that is our breeding leader in the so he's the breeder of our breeding program. And Josh, he's working on wheat, but also he's very interested in working in triticale. The triticale, probably you know this, but I'm just saying, is a hybrid between wheat and rye. And something that is very nice about triticale is that it at least in our fields here in David, they show a 40% increase in biomass when you compare to wheat, and that's translating in around 50% increase compared to wheat too in the field. So one of the reasons is because triticale has very huge spikes, so they are very big, very long. And for me, it's a great crop, or because it's a young crop with not too much breeding on that, and it has a lot of traits that can be improved, like quality. But Josh, at that moment, he was worried about plant height because the spikes are so large and so heavy that he had a lodging problem with these triticales. So basically, he told me, and we were discussing, that he would like to make the plant a little bit more compact. So that was a very good question for me because I have done some work in plant height, and I know some genes that I will show now. So we start working on that. The other thing that Josh told me, so the main genes that control plant height in wheat after the green revolutions are RHD1 genes or alleles of RHD1. Those are dominant mutation in the DELA genes. So these are point mutation or deletions in the DELA domain that will make the DELA protein insensitive to GA, to gibberellins. So basically the DELA protein is always active and repress or alter the activity of different transcription factors. And at the end, what you have is the shortest plant because it's insensitive to GA. So one of the first things that George told me is, I want to make the plant shorter, but I don't want to work with DELAs. I don't want them in the triticales because, and the reason of that is because breeders now are trying to move away from RHTV1 or alleles because they have other pleiotropic effects. I mean, they don't just reduce plant height, but also, for example, the case of RHTV1B, that is the most common allele using in wheat, it also reduces the size of the seedling. So not just the plant height is shorter, the whole plant is smaller. Shorter seedlings have the problem that you cannot plant or use them in semi-arid region where you have to plant deeper to get the humidity on the soil. So basically, there was a need to develop new or novel high loci or genetic material with less pleiotropic effect than the DELA. So I started working on that. And the way that I approach this, this was part because of my experience with flowering, and also because I was, as I said at the beginning, I was motivated by the work done in barley by Sarah McKean and Robbie, that I will consider, or I consider plant height actually as a part of the reproductive development. So it's not just plant size. It's plant height in wheat or also in barley is basically the stem elongation during the reproductive transition. That is internal elongation and peduncle elongation. And that process is part of the reproductive development. So if we want to target that process of reduce plant height, we can focus, my idea was to focus on regulators that control the reproductive transition. And one of those regulators that I work a lot in Jorge's lab, and also there was good evidence in barley, was this model that is microRNA 172 and AP2 transcription factors. So this model, microRNA and target, are very conserved in different plants. They participate in most of the plant in the transition to reproductive development. And I knew that the microRNA expression, for example, increase or is induced during the reproductive transition. So at early stages, the stages that we didn't want to modify, the microRNA expression is very low, but later increase and is active repressing the AP2 transcription factors. I also, at that moment, I knew that if we reduce the activity of the microRNA 172, this is a transgenic approach that is called target mimicry. So mimics is a non-coding RNA that can bind to the microRNA, and the microRNA cannot cut it. So the mimic basically sequesters the microRNA and is not active. So by reducing the activity of the microRNA, basically what we got were shorter plants. So that means that reducing microRNA activity can be a way to generate shorter plants. But this is a transgenic approach. There is an easier way to get a similar effect, that is by introduction mutations that affect the microRNA activity. And here I'm showing the beautiful work done in barley with the characterization of the CEO mutants that basically are these mutants can point mutation in the microRNA binding site of microRNA 172 that reduce the activity of the microRNA. So you can see here in this work. This is Houston 2013. They showed that the mutation reduced the microRNA activity, so it's less efficient in cutting this allele. And the result of that is like you can also get shorter plant. So you are not here. We are not reducing microRNA activity by the mimic, just by mutating the microRNA binding site, so the microRNA cannot bind there. A similar example I found in wheat, where I got it using the Jorge Thielen population. I found a point mutation in the microRNA binding site of the ortholog of the CO gene in wheat, and that point mutation also resulted in a reduction of plant height. Okay, so but from this Thielen mutation, the reduction in plant height was there, so the phenotype was clear, but in the level of reduction, or say the intensity of that reduction, was too low and probably not good to use that as a tool to reduce plant height in a breeding program. But this triggered my ideas or my question about if we can, by modifying the microRNA binding site of this AP2 transcription factor, if we can generate stronger alleles that have a stronger effect on plant size or on plant height, sorry. And also, but also that brings many other questions. If those stronger alleles will affect seedling development or will have other pleiotropic effect. If we can reduce plant height independently of the DELA alleles, for example. And the most important point for me at that moment was if we can incorporate these novel alleles into triticale, if there will be an easy way to move them to triticale. And again, well, CRISPR for this is perfect. And the combination of CRISPR with GRF shift allow us to test all these questions very quickly, and I will show the result that we got. So the good thing here is that in the genome B of this transcription factor in the microRNA binding site, there is a PAM sequence. So we can very easily target the microRNA binding site with the Cas9 and with the guide RNA and introduce mutations in that region. So the first question was, and this work was done in collaboration with Chausson. Chausson have done a lot of work, amazing work. So, to check the first question, we did a transformation with a vector having Cas9, the guide RNA targeting the microRNA binding site and GRF shift in Chronos. I mentioned Chronos is it has the RHD-B1B, the semi-dwarf allele, but for us it's our most efficient cultivar. So we transformed there. We were able to regenerate many independent T0 events, in this case 43. And after transferring them to soil, the T0 plant, we were able to genotype them to identify if they have or not mutations, and phenotype for plant height. So plant height is amazing, or it's very interesting trait because we can phenotype that directly on the T0 plant. So we can do that very, very quickly. So we don't need to advance generation to check this phenotype. And we basically, by sequencing, we were able to classify the T0 plants in wild types, heterozygo or homozygo by allelic on mutation in the microRNA binding site. And you can see that we were able to get a stronger reduction on plant height, basically because we got mutation that disrupt the microRNA binding site in a way that is more drastic than the point mutation that we got in Thielen. And also there was a clear dosage response because heterozygote plants were showing an intermediate phenotype. What was interesting too is that even this event that were very strong on reduction in plant height, the seedling site was not affected. So that's interesting, and that's something that we were looking for. And the way to explain this is because microRNA is not expressed at that stage. So wild type AP2 or a CRISPR AP2 will be the same. They are not seeing the microRNA at that stage, so seedling are normal. We want to test also this if this allele can reduce plant height independently of the alleles. And we were lucky because Jorge in his lab, he has a Chronos where he have introgressed the RHTV1B wild-type allele. So these Kronos plants are isogenic to the previous one, but they have the tall allele, so these plants are very tall. And we transformed that. We got again many independent events, and we were able to classify by genotyping again in the different categories like wild-type, heterozygote, and homozygote biallelic. So, and here you can see that even in RHTV1B background, the mutation in the microRNA can reduce plant height, and the reduction in plant height is actually more or less on the same level as that you can have when you have them. So this is Kronos with RHTV1B. So let's say using Kronos with wild-type DELA and editing the microRNA binding site or introducing the mutant DELA have a similar effect on plant height, so we can reduce plant height more or less to the same level. It will depend on the CRISPR line that we use. But what is super interesting is that while the RHTV1B reduce seedling size, our CRISPR line have a normal size on the seedling at the seedling stage. So, okay, so imagine that we were able to test all these hypotheses, but also we have in a single vector a way to introduce novel or genetic resources for plant height in any cultivar. And the original idea was to introduce this in triticale, which we were able to transform triticale. So you can see here. So we target in triticale the microRNA binding site, but here we target, so triticale is sex haploid, it will have genome A, B, and R. And B and R genome has the pump size, so both of them can be targeted by the same guide RNA. And after the transformation, again, in triticale, we were able to recover shorter plants. So you can see the reduction in plant height. And those plants also show no effect on seedling, so they were shorter, but seedling size was normal. But something that was interesting also in triticale was this. You can see these are two lines that we obtained, but line 151 is shorter than the other one. When we sequence them, we observe that they had the same mutation. It was a single nucleotide deletion in the B genome, but they had different mutation in the R genome. So line 134 has an insertion, but 151 has a deletion. So the prediction based on microRNA mechanism is that the insertion of one nucleotide will be a weaker mutation than the deletion. So the deletion has a stronger effect disrupting microRNA activity. So on that line that has a mutation that disrupts more the activity of the microRNA is the shorter one. So here again, you can see that there is a clear or nice dosage response to the microRNA activity with plant height. But in triticale, compared to wheat, when we did with only mutate B genome, in triticale we have both B and R genome. So we can not only play with the identity of the type of mutation that we are using, for example, +1 or -1 to control plant height, but also we can play with the combination of different genomes. And with that, we can generate a very nice gradient on plant height. So so you can see that combination of one weak with one stronger has a weaker reduction in plant height than two strong deletions. So, and is a very nice system because now a breeder, in this case Josh, can select specifically the plant height that he would like to have in the field. And this also is to show that even the plant that has the shortest height, that probably this will not be useful for the field because it's too short. But anyway, this plant that is too short doesn't have any effect on seedling development. Okay, the other thing that is interesting is that this plant here that has single mutation, USDA here now is not regulating them. So this single event, CRISPR event are exempt of regulation. So they can go to the field directly without any permit. So, and this is to show that closing the loop with Josh, we were able this year to put this plant in the field. So he wanted to make a plant a little bit shorter, and this year we were able to put them in the field, and you can see the wild type original breeding line from his breeding program. And here we have a completely isogenic line that has only a single nucleotide deletion, and you can see the effect on plant height. So we are very happy with this result. Okay. As a summary of the second part, I show you that we can use all these characteristic of feature of this technology to quickly incorporate new trait using CRISPR. In this case, we target the microRNA binding site of AP2 transcription factor that result in reduction in plant height without effect, pleiotropic effect, and seedling development. And we transfer that results into a breeding line, in this case, triticale, that we were able to test in the field. So with that, I would like to thank David, all my collaborators, especially David and Jorge. David, he was, as I said, he was the manager of the plant transformation facility until last month. He retired. Now I'm the manager of the facility. But well, David is coming to work again in a couple of weeks as a part-time, so we will still working together as collaborators. Jorge and Chausson and Josh from Jorge's lab, and also I'd like to thank Javier, that was my PI in Argentina, where I started working with CRF Shift. And all these are all collaborators that are testing our CRF Shift technology in different crops and in different, yeah, basically different crops. Okay, thank you.
Thank you, Juan. Very, very interesting talk. Really appreciate it. Now we will pass to the session of questions. I see that Robbie already had a question on the chat box. Maybe you want to ask it directly, otherwise I can read it. And if people have questions, they can just raise their hand and ask the question directly, or add it on the chat box and I can then read it for you.
Okay.
So Ruby, maybe you can ask your question.
I can read it, my question directly. Nice talk, Juan. Nice work, actually. So the question was about the mutations in AP2 and the microRNA binding sites. Maybe it's because we're on a small screen, but it didn't look like there was a big effect on spike size.
No, you know, yeah, I would say so, and this is something that we are, I would say, learning now, or with the plant material that we were getting. So there is the triticale lines. They have a little bit more compact spikes, but if you look on the, actually yesterday I was trying to get a better picture, but I couldn't find it. But if you look at this figure, the effect is not very dramatic. So they are more compact. But they look quite okay, normal. But in Kronos, for example, the phenotype on the spike compactness is stronger. So the spike became much more compact. My idea or my hypothesis is that so the effect on the spike compactness will depend a lot on the background, and triticale, initially the triticale line that we were using has a very lax spike. So I think it was probably a combination of that background that we were able to make it shorter, but because it was lax already, because of the genetic background, the effect was not that big. So what I'm thinking is like if I'm going to use this strategy to reduce plant height in another with cultivar, I will select probably with the line that has a more lax spike to get the less pleiotropic effect on spike compactness. I mean, I show, but we were working with Josh and other people with the Kronos line. The spike is super compact, but they say it's fine. I mean, they say it's okay. It's compact, but it's not a big problem.
So there's no reduction in seed number per spike or seed size?
No. What we observe, the experiment in the field, we harvest that experiment like 3 weeks ago. So we don't know yet how will be the yield. Even so the experiment in the field is not perfect. We would like to repeat another year with plots to check yield. But this year we did rows, so at least we will get a feeling on plant or row or per plant. We don't see in greenhouse, for example, an effect on seed number. Spikelet number, for example, is the same. So there is no reduction in spikelet number. And what I observed, that I think in barley you have the same phenotype. The grains are a little bit more rounded, more shorter and rounded.
Mhm.
But in triticale, that happened only in the strongest line, not in the intermediate one.
Okay, thank you. I see you have other questions, so we better get off the floor.
Okay.
Thank you. There's a question at the chat box from Yunshin.
Yes.
She said, first, very nice talk. And she's a little bit confused about your AP2 CRISPR mutant.
Yeah.
Are they knockout mutant, reading frame change, or they will just the opposite of the MER2 target side mutation,
Yeah.
which create overexpression of AP2 actually?
Yeah. This is a very good question. And that was a question that I have also when I started, because probably the best strategy would have been to do base editing, so to modify the microRNA binding site without changing the reading frame. But let me see if I can show you that here. Can you see that?
Yep.
Okay. The microRNA binding site on this AP2 transcription factor is at the very end of the C terminal. And this is AlphaFold prediction of the AP2 structure. The microRNA binding site is here. So, and also if you look in wheat and barley RNA-seq data, you will see that there are sometimes alternative splicing that will remove the last exon that had a microRNA binding site. So I have some evidence and data that if we change the reading frame here by disrupting the microRNA binding site, probably will not have a strong effect or no effect on the protein activity, because it's after the AP2 binding, AP2 domains and very close to the C terminal. And actually, when I show you here in Chronos, for example, let me go here. We have in our CRISPR population, we have mutant that had deletion of 1, deletion of 2, or deletion of 3. So deletion of 3 will remove 1 amino acid but keep the frame. And they all have the same phenotype. And we have, I'm not including, but we have also deletion of 6, deletion of, I think we had deletion of 33, deletion of 15. So there is a very large combination of mutations. I mean, we cannot discard that part of the phenotypes on this allele will be because we are changing a little bit the protein. But I think that it's more related to microRNA activity because of all this evidence that, that you can see that different alleles, even the one that keep the frame, they have exactly the same phenotype. We have some that, a few alleles that are amino acid changes, but those are weaker. So those look like the TILIN mutants. Again, you can predict that, but based on microRNA activity, that point mutation will have a minimal effect. So the point also, I don't know, I wouldn't like to make big claims about the protein activity, but for me at this moment was more like a tool to introduce plant height, and it's working. So it's producing what we want. Maybe we'll need to study a little bit more about the protein activity later. Okay.
Okay, thank you. Sarah, and don't know if she wants to ask her question herself, or I can read it. Did you observe any change in the logical development with this AP2?
Yes, I didn't check in the CRISPR, but in the TILIN population that I show you, those TILIN population, they have a reduction in the logical size. So the same phenotype as you have in barley. But the reduction is interesting because it's half. Let's say it's because you still have the genome A being active, so it's very dosage dependent. We didn't check. I mean, I didn't check in the CRISPR, but I assume based on the tilling that they will have the same reduction on lodicle size. Yes. I think, Sarah, I remember many years ago that we met in PAC, that in that meeting I present the MIMIC 172 results. And I remember that you asked me that, or we were talking about the same question, the effect of MIMIC 172 on lodicles. To me, that's a very interesting result. I don't have a good explanation yet, but the MIMIC doesn't have an effect on lodicle size. So I don't know if that's because the MIMIC is not active on lodicles, or because the MACE ubiquitin promoter that I'm using to drive the MIMIC is not expressed in the lodicles. I don't think so, but MIMIC 172 has normal lodicles, but the point mutation in this AP2, yes, they have.
Thank you. So there is another question here. While screening the plant for mutation, is it possible that the plant is positive for Cas9 but not genome edited? That will be what could be the possible reason?
When we have a positive plant that doesn't have edit, but is Cas9 positive. I think that depends a lot, in my experience, depends a lot on the guide RNA. So there are guide RNA that maybe are not very efficient, and you have a larger frequency of wild-type plants. It's like maybe the insertion site, the expression level of Cas9 is weak or low, combining with the guide RNA that is not very active, so that gives you wild-type plants. For this guide RNA, I will consider it like intermediate, because we were able to get a good number of homozygote or biallelic plants, but you can see that we get also a lot of heterozygote. So even in those plants that we were able to edit one chromosome, the other was wild type, so it's not 100% active. Yeah, but I will consider that that's probably the main reason, that it will depend on the guide RNA. For example, for gene Q guide RNA, that guide RNA is super active, and 100% of the plant have editing. I forgot to connect. I'm getting a message. I don't know. Sorry. I'm running low on battery because I think I'm using the wrong. I will connect.
Okay.
I plugged my computer, but probably I'm using the wrong one. Let me see. Now it's working. Yeah.
Okay.
Sorry. Yeah, that's. I think that's my probably my conclusion. That and sometimes I transform guide RNA that are not active at all, so I'm not seeing any activity, and all plants are wild type. So.
Okay, thank you. I see Wendy open her camera. Maybe she may want to ask her question directly.
Hello there. Hello, Johan. I can do. It was just I'm interested in sort of wider application of the GRF GIF system. And you gave that nice data in maize, and obviously you mentioned at the end it's been tried in a wide range of species now.
Yeah.
I'm just wondering, particularly for this audience, if you have any additional information for the use in barley, and whether you can maybe say something about the wider application.
Yes, thank you, Wendy. That's great. I was planning to have something, but it's still very bad. I have planned, but it's preliminary, so I didn't have time to include them. But I was working in collaboration with Sebastian to do. So the idea was to mutate a family of genes in wheat, and Sebastian was interested in barley. And also I wanted to incorporate, I'm incorporating the barley service also in the transformation facility. So I started doing some experiments in Golden Promise. So my data will be Golden Promise, and so I haven't done anything like crazy going to more difficult cultivars. But in Golden Promise, and also I talked with Sadish about her experience on barley. She told me that she was getting a lot of calli, but the calli was having problem to regenerate. But in my hands, and I'm following your protocols. What I observe is that it's critical the time that I have the calli in media with cytokinin. So when I reduce that to the very short window, I'm able to regenerate a lot of plants. So for Sebastian, I was able to, in a single experiment, to get a lot of plant, even using barley plant that I'm ashamed because the the quality of those plants were terrible. But I was able to get a lot of plant. But it was critical, and I had to play a little bit with that to reduce the timing that I had the calli in cytokinin. So in your protocol, you have I think it's 2 weeks with BAP, and then you move to the regeneration media without hormones. So I play a lot with the dosage of BAP and the time that was there, because my point is that if you keep the calli in BAP or also I tried C18 longer, it goes to a different pathway. It will get you transgenic calli that it grows like a crazy, but you cannot get plant. But reducing that, I was able to get a lot of plant. Yeah, like it will be like the wheat system. Golden Promise. I don't have experience with other cultivars.
No, that's really interesting. Thank you. Actually, we've recently the step that you've mentioned with the BAP, we don't actually need to include that anymore. So maybe we need to go back and have another look. But yeah, no, that's interesting. Thank you.
I would say that also with C18, I got a very nice response, but C18 even I had to reduce a little bit more the timing. But I observed that C18 was a little bit more active than BAP to promote the shooting.
I don't see other questions in the chat box. I may have one question. Actually, I really enjoy your approach to adjust the development of the plant height by rather to make a loss of function or removing the microRNA to just play on the target side, and you show, at least as far as I could see, no pleiotropic effect. I thought you had the PAM motif, that PAM site that led you to target. In absence of a PAM site for another microRNA and target gene, do you have other strategy or other Cas protein that may target other motif, and so to kind of explore?
Yes.
Yeah, to play with this more.
Yeah, I mean this case was actually very simple because it has a PAM site and because the target site was close to the C terminal. But for most of the other microRNA targets that, I mean, we work or we know, the situation is not like that. I mean, SPL transcription factor, GRF, TCPs, all of them had a microRNA binding site in the middle. So in those situations, yes, probably I will use a base editor, or the strategy also I have in mind is like, because we can regenerate so many plants that just by chance we will get 3 nucleotide deletions or amino acid change using the regular Cas9. And the PAM sequence, we are working on testing alternative of Cas9 that have different PAM recognition sites. So that's also something that we are doing, yeah, in case that you want to go. But I know the SPL binding site for several, the microRNA 156 binding site for several SPL, for example, has a PAM site very nicely there that to use, GRF the same. So I think there are many opportunities to do stuff like that. Yeah.
Okay, thank you. I think we have no more questions. Last chance, maybe for last one for people, or otherwise, I think it will be finish for the talk today. Okay, thank you, Juan. I think your kids are awake. I can hear them.
You
So
hear
you're in
that?
time to take your breakfast with them.
Yeah, no, I'm happy that they didn't came and open the door. So they are doing well. They're just playing downstairs. Okay.
Thank you for the talk today. It was very interesting, and I'm sure everyone enjoyed that you presented for us.
Yeah, thank you for the invitation. And so I say that now I'm the manager of the plant transformation facility. I want to incorporate barley as one of our crops. So I will be happy to collaborate and work with any that is interested in doing stuff in barley. So I think that we have an opportunity to do this kind of experiments, like are more like I would not say high throughput, but we can work more like with big numbers, a lot of results that I mean, that's the way I used to work in Arabidopsis. So I feel like right now we can do this kind of work in wheat and barley, like before I was doing in Arabidopsis. Okay, well.
Thank you. Thanks, Juan. Great
Thank
talk.
you.
Thank you, Juan.
Thank you. Bye-bye.
Bye-bye.
Bye-bye.